Joining me today is Dr. Anna Dünkel from the Fraunhofer ICI, the Institute for Cell Therapy and Immunology in Leipzig. Thank you, Dr. Dünkel, for joining us today.
Yeah, thank you for having me. I am happy to be here.
The pleasure is all mine. Now, for everyone not familiar with the Fraunhofer ICI, could you perhaps briefly introduce what the Institute does? I'm just going to make you moderator.
Okay. Yes, die Fraunhofer Gesellschaft is headquartered in Germany, and we are the world's leading applied research organization. And here in Leipzig at the ICI, we are working on different kind of technologies and ideas, but it's all around cell therapy and immunology and vaccines, for example. We are working with therapeutics from the ATMP spectrum. And yeah, this is what we do here.
Thank you. Now, anyone who's been following the research and development in oncology in recent years has pretty certainly come across CAR T cells at some point. Now, a part of your research is about CAR NK cells. Could you tell us a bit about what their characteristics are and what sets them apart? What are the differences and potentially the advantages compared to CAR T cells?
Yeah, so if you want to talk about chimeric antigen receptor NK cells, you always have to talk about CAR T cells and the challenges in CAR T cell therapy. And I will just jump right into it. And one of the biggest challenges is that the production costs for a CAR T cell therapy are pretty high. We are here about $350,000 per product. And there are also long processing and manufacturing times. On average, we're talking about 3 weeks and above from leukapheresis to infusion of the CAR T cells. And about 10 to 15% of the CAR T cell therapies cannot be given since the patients present with a poor health status and or die because of the fast progression of their disease. Since it is an autologous cell transplantation, the quality of the patient T cells is quite challenging at some point because they are suffering from cancer and often show impaired T cell function or decreased T cell counts, which is also challenging in terms of production. And up to 30% of Kymriah products don't meet the release criteria from the FDA, which is the Food and Drug Administration from the US, and are no longer available then for the respective patients. Yeah, and the last note, and really important, is that CAR T cell therapy is associated with life-threatening side effects, namely cytokine release syndrome, but also neurotoxicity. So all these challenges are there, and to find another way of addressing the therapeutic need, manufacturing of CAR NK cells was developed. So when you have the NK cell here on the left-hand side, you can deliver a certain gene, which is the so-called CAR, a chimeric antigen receptor, which is then expressed on the cell surface. And when you have these cells, they have to be expanded because you need a certain amount of them or a certain cell count. And then you can cryopreserve the cells. And this is something which is quite nice because you can use allogeneic transplantation settings. So you have a master cell bank and then can use the frozen cells off the shelf. That means when there is a patient, you can just thaw the cells and then apply it to the patient. So these the cells then are continuously available, and the production costs are significantly reduced. You find different numbers here, but round about $20,000 per product. And Yeah, since NK cells or natural killer cells have limited lifespans, they also induce minimal toxicity against healthy cells, which express the target antigen. And you also have a reduced risk of GVHD, which means graft versus host disease, which is something you can also see from stem cell transplantation. And also a reduced risk of side effects is observed, which means the cytokine release syndrome and neurotoxicity. And other than T cells, you can give them repetitively. So you can give one dose and then another dose on another time point.
Ah, that's interesting. Now, you already mentioned the factor of the possibility of using cryopreservation in the case of CAR NK cells. Is there anything else you can tell me about the manufacturing and general handling and the characteristics of CAR NK cells here?
Yeah, this is a little bit different from the T cells. You can use for the manufacturing, you have different kind of sources you can use for the start of your production. You can, here on the left-hand side, use primary cells, or you can also use cell lines, since the NK cells can be used in an allogeneic transplantation setting. So you can use cells from a different person than your own cells, which is now the part of the CAR T cell manufacturing. So from the primary cells, you can maybe use cord blood or also peripheral blood from healthy donors, for example, or stem cells. And as I mentioned earlier, you can also use cell lines. And it depends what kind of source you use, then you have advantages and disadvantages to it. So I will just go straight to the production process. As you can see here, it is depicted an healthy donor. And then, of course, the availability of the cell material is great, and also the transduction protocols for primary cells. So transduction means you take a virus, and the virus will carry your gene into the cell, which then leads to the expression of the receptor on the surface. So, and these protocols are improving constantly, since there is a therapeutic need for this kind of products. But on the downside is that you have a donor-dependent variability in cell numbers. So not every person has the same cell numbers as the other one. So you have 5 to 15% of NK cells in your blood, and yeah, it is really donor dependent. And of course, you can reach higher transduction rates for cell lines, which means that you can deliver your gene of interest in a more efficient way in cell lines. And of course, since they are primary cells, they really don't like to be frozen and thawed 100,000 times. So you really have to think about the process development and when to put your freeze and thaw cycles in.
Ah, well, you already mentioned some challenges associated with it, such as the donor-dependent variability. And are there any other challenges that you in particular are working on?
Yes, so our focus is mainly on the gene delivery, which is, oh no, yeah. At this point, when you take your DNA and put it into the cell, that the cell will express your protein on the surface. So there are different kind of protocols to do it. You can use different kind of DNA, but also RNA, and you can maybe just shock the cells with an electronic, with an impulse of an electroshock. Or you use the virus, as I said earlier. And yeah, it depends how the design of the gene delivery is really. So you can work on different parts in this protocols. And then another thing, we're working on the freezing and thawing too, because there are also automated solutions to freeze and thaw the cells, which are under consideration for a further manufacturing protocol. And then, of course, we are working here at the Fraunhofer IZI at different kinds of plasmid designs for the gene delivery. And yeah, so we play around a lot and try to find the best things for certain kinds of products in the end.
And concerning the application in clinical practice, in which settings, clinical settings, do you see the highest potential for CAR-NK cells?
Yeah, this is a good question. I will jump just right through my other slides and show you this one. So here I try to focus on the clinical trials going on at the moment, and you can find these informations openly on clinicaltrials.gov. This is nothing too secret here. So you can see the numbers of the clinical trials for the solid tumors are not too many, but I think solid tumors are always discussed in this whole cellular therapeutics area, because you have these cell mass. And of course, if you use a cellular therapeutic, then it will come from the outside. But what is inside the tumor mass, you cannot really control. So there are lots of different ideas on how to overcome these obstacles. And yeah, you can also see here that in the clinical trials, they used different kind of NK cell sources, cord blood or peripheral blood, and also cell lines. So it really is a lot of going on. But I think for the solid tumors and for combination therapies, which means established antibodies against different tumors together with CAR-T and CAR-NK cells, I think this could be the next step in the future.
That's really interesting. Thank you very much, Dr. Dünkel, for sharing these insights into your research work.
Thank you for having me. It was really nice.